neutrophils, neutrophils will recognize the surface, which is coated by the immunoglobulins and the complement, and you get opsonophagocytosis. Next slide, please. Next slide, please. Yeah. So what can bacteria do to prevent this opsonophagocytosis? The first thing is bind to fibrin and generate actually a fibrin clot around you. And this is, of course, a big problem because if you have a wound, fibrin will try to stop leaking. Well, as aureus can bind to it, and in the top, in A, you can see in yellow the bacteria, and in blue the fibrin, that the bacteria are totally covered by this fibrin. B is the bacteria alone, and C is the fibrin. And then D, you can see a magnification of the bacteria in the fibrin clots. Next slide, please. Another way to prevent opsonophagocytosis is getting a big capsule. The problem with a lot of these capsules, polysaccharides, is that they are very low immunogenic. So we don't generate immunoglobulins often against them, making it very difficult for our neutrophils to recognize the bacteria. And you need to really vaccinate, for instance, actively in order to get antibodies against, for instance, the pneumococci. And you all know. Please continue. Next slide. Incorporating into bone. And I hope you can see the bugs sitting there incorporated into the bone. Thank you. So it melts or it incorporates and it sits there. And as you probably understand, for an orthopedic surgeon, it's very, very difficult. You really need to scrape it out. Otherwise, you will not be able to get it out. Next slide, please. And that this is really an issue is this slide. On the top left, you can see in green bacteria. And of course, these are biofilms, so there are 100 or more in one of these blobs. In red, there are the neutrophils. And when you look after a half an hour, nothing happened. And that is the slide, the picture underneath the top one on the left side. So the neutrophils really cannot handle these. And is there also an appointment? No, it doesn't work. Yeah. Yeah. But oh yeah, yeah. So here you can see the bacteria in green and the neutrophils in red. And after half an hour, nothing happened to the bacteria. While phagocytosis works very, very quickly. I'm not sure if every one of you saw this. Within a couple of minutes, you will see phagocytosis if the bacteria are opsonized. So bacteria in a biofilm, in a cluster, can escape the opsonophagocytosis. And not, oh. There is another problem for bacteria the moment they get into our body, and that is antimicrobial peptides. In all our liquids, antimicrobial peptides are present. And they are like soap a little bit. They are predominantly cationic and hydrophobic, which is, of course, like a soap. And they are in our tears, they are in our saliva, they play a big, important role in our intestinal tract to keep the bugs at a reasonable level. Otherwise, we would be overwhelmed by the bacteria. So these antimicrobial peptides, let me have a look if I can. Yeah.
I think it doesn't really work. So.
Anyway, in the middle, you can see the antimicrobial peptides interacting with a bacterial membrane. And bacterial membranes are slightly different in comparison to our membranes because they contain a lot of phospholipids, which makes them rather negative. So these cationic antimicrobial peptides can interact with the... Oh, you try to get the... Yeah. Oh. Can you please give me... I will try to get... Yeah. So the moment the antimicrobial peptides interact with the lipid layer, several things can happen. And this is depicted in this picture. In the left side, they form a channel. On the middle, you can see also a different form of channel. And on the right side, you can see that they can form, depending on the concentration and the interaction with the bacterial membrane, they can even form micelles. Bottom line is, the bacterial membrane will be damaged, which is, of course, a big problem for the bacteria. Yet, if it wasn't for the bacteria, and bacteria are no-sitting ducks. And what you can see in the left top, this is, of course, a scheme of how Staphylococcus aureus looks like. You see the peptidoglycan in the top, and then the membrane in the middle. And if an antimicrobial peptide enters this surrounding, in the left, you can see a two-component system that can interact specifically with this antimicrobial peptide. And then the response of the bacterium is, and that you can see in the middle, that the wall-toleric acid, which is part of the cell wall, incorporates alanine. And alanine is positively charged. And so by sensing antimicrobial peptides, the bacteria can change charge, become more positive, which is, of course, a problem for the antimicrobial peptide because the antimicrobial peptide is also positively charged. So you get repulsion. As I already mentioned, in the left, you can see here the phospholipids of bacteria are quite negative. Another part of the reaction of this two-component system is that lysyl groups, which are also positively charged, become incorporated into the cell wall, or cell membrane, I should say. So if the bacteria senses the antimicrobial peptides, it becomes more positively charged, causing repulsion of antimicrobial peptides. This has a consequence, and maybe this consequence was already known by you. A lot of our antibiotics are positively or negatively charged, or sweeter ions. If it is positive, so if the bacteria enters the human body and becomes more positive, this has a consequence for a lot of antibiotics. Some will be repulsed when they're positive. If they're negative, then they will accumulate in this layer, and you need to add a whole lot more in order to kill the bacteria if the antibiotic needs to get into the cell. And then, of course, the sweeter ions can have a problem. They are positive and negative, so the positive charge will stick to the negative, and etc., etc. So the natural response of the bacteria towards the immune system has a consequence for treatability for possibility to work with an antibiotic. And this picture I don't have to actually show to you. You know this better than me, how antibiotics work. But I would like to stress here that a lot of these antibiotics need to enter the human body, of the bacterial body. The bacterial cell. And besides the change of charge that I just mentioned as a consequence of antimicrobial peptides, as Staphylococcus aureus can generate an extracellular polymer substance which contains several molecules, often anionic or cationic charged. For instance, extracellular DNA, which is of course negatively charged, uronic acid. On top of the charge that I already mentioned, the bacteria prevent, in a biofilm, are preventing antibiotics to penetrate into the bacterial cell. Furthermore, for antibiotics to work, a lot of them, antibiotics, work only on dividing cells. And it has been known for quite some time that a lot of these bacterial cells in a biofilm are less active. So looking at a planktonic, nicely dividing group of bacteria, or looking at a biofilm, you have very different state of metabolic activity, making them, on top of all the things that are already mentioned, again, less sensitive for antibiotics. It has been mentioned that in comparison to planktonic grown bacteria and biofilm associated, up to 1,000 times less sensitive bacteria are for antibiotics. So this is more it was more an introduction. How can we approach this? How do we look at this? Well, this will activate some problems, I guess, with Dr. Sultan, because he worked for 1 year roughly on this essay. It's a very bad essay. I think he will confirm, and he said afterwards, "I will not do this anymore." Huh? Yeah, there you go. So what do? You grow a biofilm in a 96 well plate, and you start staining them with crystal violet. I can tell you, don't try this at home. It's irreproducible. You wash away very quickly the bacteria, and it's still the most used assay. So one of the biggest problems here is that you not look at bacteria, but you basically look at the proteins that interact with your crystal violet. So what on earth are we looking at? Huh? Don't say this loud, because most people working in biofilm work will use this, and they will kill you. Huh? So I'm trying to make contact. It changes over here, but it's not changing over there. Maybe I should. Can I?
I don't think the cursor will be work on that computer because the main host is here. Maybe you can just continue, sir. Yeah, it's not possible to activate the cursor there.
Okay, then
Bisa Pak Taufik? Apanya?
Putus ini.
Ada masalah dengan komputernya? Hostnya?
works again.
Okay. This
Yeah. Oh, sorry.
Yeah.
But also to move. I'm not sure how to.
You just if you click something, you can click again to the screen to the pen. Yeah.
But I'm not sure how to move now to the next slide.
Yeah, you can just click it.
Okay.
Click yeah.
So how can we quantify the impact of antibiotics on biofilms in a reasonable and reliable way? A couple of years ago, a company asked me, hey, we developed a machine. Are you willing to test it? And then I said, quickly, yes. Because what they offered me was an isothermal microcalorie meter. And until then, I knew what microcalorimetry was. But when I was a student, these machines were room filling. Yeah? So this, and you only had one sample, and you can could go to the chemical department, they put in a sample, and then if two molecules, for instance, interacted, and it would take energy, then the machine could determine, hey, I see a difference in energy being performed. This machine has 32 wells that can simultaneously determine is there energy being produced, yes or no. The good thing is it's very, very sensitive, as you can see here, how sensitive it is. So roughly 10 to the power 4 bacteria are already generating enough heat to be determined. So, what did we do? We started growing bacteria. And this is actually one of the first experiments that we did. And I hope you can see that it looks really like a nice growth curve, exponential growth. The trick here is that we didn't use bacteria media. And actually, I wasn't performing this experiment, but Dr. Sultan did. And one of my technicians, Mary. And what we could see is that the slope at a certain moment stopped over here. And then very slowly went down. But it still produces heat. So we wondered, could it be that we are looking at biofilms? So we took out the vial and look, here they are. Small colonies. this polyp forms already after 24 hours. You can see them with your bare eyes. Huh? And when you put them back into the machine, you can also see how much heat they produce. So that's a very nice measure of active bacteria. And then what we did, we took the media out, we washed it, and added new media with antibiotics. Look over here. This is the untreated, and this is the treated with rifampicin. And then you can see that after already 1 hour, because the machine is so sensitive that it takes 1 hour to establish itself, and then it can start measuring. But already after 1 hour, you can see that the rifampicin is really giving the bacteria a very hard time. Yeah? There's also one other thing. Sorry, then we started calculating how can we do this best. And in the end, we did this. So we normalized for the negative control, the growth control. And that's seen on the right side. But there is something disturbing can be seen in the picture on the right side. It goes up again. I will come back to this later. Oh. No. We took a collection of MSSAs and MRSAs. The MSSAs are depicted in the left side. We ran the cells through the Vitek. We also did microdilution assay. So this is the MIC. So here you can see nicely the MSSAs, which are very sensitive all to rifampicin. And these are the MRSAs with one exception. All strains are sensitive for rifampicin, except for this mu50 sitting over here. So what we did is we took the resistant strain also in this pattern, and you can clearly see that the strains that are sensitive really have a hard time, though two over here start growing again. When we took them out and put them in the Vitek, they were indeed resistant. And that's, of course, what you know. Huh? Simultaneous monotreatment with rifampicin causes development of resistance. These are the MRSAs, and we don't see this in this group of MRSAs happening. So most cases, the MRSAs remain sensitive. And now, I hope. Another thing, in my country, MSSAs are usually treated with Fluclox. We treated on the right side here, the MRSAs are all resistant, as you expect, Vitek, but also in the MIC values for rifampicin. And when you look at the profiles that we get from the microcalorimeter, you would expect this. Everything remains more or less unaffected. But what is disturbing, the MSSAs, and we put in over on the left side again the mu50 as a control, which is resistant. I hope you can appreciate MSSAs in a biofilm are not sensitive to Fluclox under the conditions tested. Another thing, vancomycin. In the US, I know the Staphylococcus aureus MRSAs are by definition treated with vancomycin. The mu50 is an intermediate resistant strain. I don't see any difference over here in our assay on in our biofilms. Not for the MRSAs, not for the MSSAs. So under these conditions, these bacteria are not sensitive to vancomycin. We added several other antibiotics. based on advice from our medical microbiologist. And here you can see in the left are MRSAs. This is an MRSA, one of the MRSAs, and actually this is an MRSA coming from this country. Huh? Dr. Sultan brought him himself to our department, and it's a very resistant strain, as you can see. And the antibiotics for which the strain is resistant. Oh, I don't see my cursor anymore. Oh, I want to go back. On the right side, you can see that the antibiotics for which the bacteria is resistant are horizontal, but the others, there is some effect, yet they grow out. And on the left side here, sorry, that's what I wanted depicted over here. So these are the antibiotics towards which this strain is resistant, and this is supposed to be sensitive. Well, they can tolerate it. And actually, after some time, they just started growing again. This is another strain, and here, this is an MSSA, which is sensitive towards rifampicin, but it became resistant after a while. And here you can see the different classes of antibiotics where there is an effect, but they don't kill them. The concentration of antibiotics used in this assay are the maximal concentration that is allowed in humans. Yeah, so this, the story is not only sad. Here we started using in the 2 strains that became resistant towards rifampicin. We combined it with Fluclox, with vancomycin, and with what was it? Levofloxacin. And what you can see is that if the strain is with rifampicin or with the other antibiotics single treated, then the patterns are similar, as we already saw. Yet in the green, you can see that if you combine the antibiotics under the conditions tested and for the time tested here, this resistance doesn't appear. Please take in mind that we only assayed it for a little more than 24 hours. Yeah? But double treatment under these conditions looks as more positive than single treatment. There are also combinations that you don't want to use. This is clindamycin and rifampicin, and the clindamycin is given in red, the rifampicin is in blue, and when you combine it in green, then the curves start being similar to the clindamycin alone. Yeah, so clindamycin reduces the effect of rifampicin for these strains under the conditions tested. Yeah? Okay, I hope I convinced you that in biofilms, MSSAs can become MRSAs. They can tolerate Fluclox. Biofilm can tolerate quite a high concentrations or the maximum concentration possible of vancomycin. If you combine rifampicin with Fluclox, vancomycin and levofloxacin, you can prevent, under the conditions tested, formation of resistance. Rifampicin combined with clindamycin shows the opposite. It's antagonistic. Now I need some more. Yesterday evening, we already talked about redundancy. I always say redundancy, if you see redundancy, it matters to the bug. What does redundancy means in this case? Bacteria have an answer, different answers for the same problem. So if you cannot work in one way, then they do it in another way. Huh? Or as we say in Europe, there's different ways to roam. Huh? So a biofilm of Azorius can look like this. And then we talk about polysaccharides in their surrounding. And that is usually an acetylglucosamine. And polyacetylglucosamine looks a lot like humans. Furthermore, it's slightly positively charged. There we are again. The bacteria can also cover themselves with DNA. Another possibility is fibrin. I already showed that. Furthermore, and that's given over here, prions can form, that the bacteria can produce small molecules, PSMs, and they form a very rigid cluster around the bacteria. So this is trouble. When does the bug do what? Under which conditions? Is it possible that in blood you can only find this kind of fibrin-coated bacteria, while under other conditions they cover themselves in another way? Yeah? I wouldn't be surprised, but we don't know yet. And so we need really to figure out how that works. And this is a nice experiment done by Dr. Sultan. TSB is, of course, tryptic soya broth. And when you stain the bacteria, which is done over here with live death stain, as we say, the living bacteria are green, the dead bacteria are red. And when you add a lectin, which binds to the polyacetyl glucosamine, then the bacteria will turn blue. In this bacteria growth media, even after 24 hours, you can see a nice biofilm formation, but no N-acetylglucosamine. When you take IMDM, and IMDM is a tissue culture media where human cells can grow in. Human cells cannot grow in a TSB, by the way, huh? But they can grow in IMDM, of course. Bacteria can grow as well. And they form these nice clusters and look at the same strain. It starts to produce this PIA, this N-acetylglucosamine, which has, of course, a different and enormous effect again, because it's charged on antibiotic sensitivity. So it really matters how you look at, under which conditions you look at biofilms, because they can be different. In the early, some eight years ago, Dr. Sultan got some money from Dicty to work in my lab after presenting his masters in my lab. And what we decided to work on is how do bacteria protect themselves during the early stages of biofilm formation? Because over there, they're vulnerable. And you see only single bacteria sitting. The layer is not yet developed in a proper way. So they cannot, so they should be able to be killed. Well, what we did, or what he did actually, is he grew the bacteria for 3 hours, took off the supernate and added new IMDM, and then took this sample and we went to the mass spec. We have a nice mass spec facility, and then you can determine which proteins are being produced by the bacteria. And this is an overview. And I hope you can see it. One of them is nuclease and the other one is skin. And skin is an inhibitor of the complement, chemotaxis inhibitor. So already after 3 hours, so in the 4th hour of development of biofilm, the bacteria starts to produce immune modulators. And what does this mean? I would like to go back to this slide again. So here are the bacteria. Where's my cursor? Here. Here are the bacteria. And as a consequence of complement activation, a gradient of C5a will be generated, which is a signal for the neutrophils to leave the blood vessel. Well, what we did is again, we repeated the same experiment and then looked at different, we added a serum, and then we could see that depending on the presence of skin, the C5a concentration decreased. Oh, sorry, I'm talking in the wrong way. The presence of skin caused a reduction here, a reduction because in the wild type, the skin was being produced, a reduction in comparison to the knockout of skin. So the bacteria is playing with the concentration of C5a that's being produced. Then we wondered, what is the consequence? So a biofilm was generated over here, and then serum or skin was added. And in this case, it wasn't, skin was not added. And here you can see that the neutrophils become activated and it becomes messy. When the skin is present, the neutrophils are there together with the bacteria, but it looks calm, which is rather strange. Yeah? The neutrophils should be activated. So, depending on the presence of this immune modulator, our immune system can be tempered, which is, of course, disturbing. Yeah? Now I would like to look at the right side of this part, the netosis. We found that already after 3 hours, the bacteria starts to produce nuclease, which is basically DNase. So over here, on the left side, you can see an incubation of neutrophils, and the neutrophils are here given in green, and they're the living neutrophils, and the dead neutrophils are in red. When you add this neutrophils to a biofilm of Azorius, you hardly don't see anything. Yeah, you see some neutrophils becoming active. But when you knock out the DNase, suddenly there is this big smear of DNA, which is netosis. That means that in the wild type, where the net nuclease is being generated, already before you can even see the nets being formed, they're already killed by the nuclease being produced by these early stages of biofilm formation. So, and this is the complemented mutant, where, of course, you should be able to see on the left. Come on, no, yeah. Over here, you can see the neutrophils, the complemented knockout. So, bottom line here is that nuclease can produced already during the early stages of biofilm formation, can degrade the nets. Again, allowing the bacteria that are forming this biofilm to prevent being killed. And I think I just said this. So the natural enemies of Azorius can, in the early stages of biofilm formation, be circumvented, the killing effect, by generation of skin and DNase. There's actually more immune modulators being produced. Until now, we didn't study this. I don't have the money to do this, but well, this is of course interesting because there's more immune modulators being produced. Some water, please. I'm good, thank you. Now I'd like to switch over to endocarditis, and this is work in progress, but we already have some very nice data. I'm working in a medical microbiology department, so we get samples, and we get samples also from heart valves that have been extracted because of an endocarditis. And this is an example of them, and I never saw it. I guess you did. If you take a sample and then send it to our department of, what is it? pathology. That's right. They took a cup, they put it in a carrier and made slides. This is slide 31, for instance. And here you can see the collagen of the heart valve in the middle. Oh. In the middle, and on the right side, on the left side, you can see the bacteria, and then, of course, the fibrin coat. So what did we do? The two slides underneath this, because they are cut like a loaf of bread, yeah, were sent to mass spec. These are the human proteins, because, of course, you get a lot of human proteins. Yeah? That's, of course, because most of the tissue is, but 1 in 100 were bacteria proteins. And this is the list of bacteria proteins being produced in vivo, yeah? Which is, of course, what we want to know. Because I don't care if they grow in tryptic soya broth. I want to know what is growing in vivo. So we did a couple of analyses, and we looked to bacteria that are growing in a bacteria media, log phase, but also the classic biofilm formation medium that is used in literature, which is not IMDM, but it's a rich bacteria media added, and then glucose is added. We also ran from these samples mass spec analysis, and here you can see that they don't look like the material that is being generated by the bacterium in vivo. And this is a list of the molecules that we see primarily being produced in vivo under the conditions. I'm not sure if you know this, but yesterday I already talked in the evening with Dr. Sultan about this, and he said, is leukocidin being produced? Yes, it's the second. So PVL, maybe for the well-known, PVL is also being found. Alpha toxin, which is a hallmark of Azorius, which kills red blood cells. Here, it's being produced. We are currently working on, because there's one problem. I ordered plasma, which is not easy in my country, but we started growing the same strain in plasma, and you know what? These molecules are not produced. So we need to add something to the plasma in order to activate this immune regulatory molecule, because leukocidins are what they are. They kill neutrophils. Wait a minute. So they produce in plasma something that is, because in this layer of vegetation, you can see the bacteria, but in an outer zone, you can see the neutrophils sitting. So there is an area where these neutrophils are not there. Presumably, they are killed by the leukocidins. Yeah? So it looks like a trench war. Yeah? I don't like war, by the way. Yeah? But it looks like a zone where the bacteria are just keeping the neutrophils out. This week, we will start working on, because we found a nice way to monitor this, and I will not go into it now, but I can, if you want, at another moment. Can we determine alpha hemolysin under the conditions in plasma? What do we need to add? And one of the molecules that is described in literature, for instance, to activate alpha toxin, is Fluclox. And these patients are treated with Fluclox, of course. They are MSSAs, and in my country, people with an endocarditis are pumped full with Fluclox. So that's the first one. There are a couple of other molecules that we think of. For instance, antimicrobial peptides being generated by the neutrophils. The lysis of neutrophils causes also antimicrobial peptides. Could it be that these antimicrobial peptides activate the bacteria to produce these toxins? Another thing is the glucose, but then the phosphorylated glucose. Because this phosphorylated glucose is a sign of interest of molecules that are interested in the cell. And Azorius is also known to activate alpha hemolysin when it's exposed to phosphorylated glucose. So we have a whole list of molecules that we're going to test and to hopefully be able to find, because then we going to test these conditions and see if under these conditions, what is the sensitivity for antibiotics. And because then we have a very nice mimic for an endocarditis. We're not there yet, but we're reaching this point. And then, of course, using the calorimeter, we can determine the sensitivity for antibiotics, etc., etc. So this is what I would like to show you on endocarditis. No, I'm sorry, I'm a little bit too quick. This is our setup for our endocarditis. But now in vitro, we grow the bacteria for 24 hours and then use plasma and expose them to drugs. And then, so here is the profile after, and this is a little bit after 24 hours in plasma. This is the profile because several times we wash them to allow the bugs to accumulate. And then this is the exposure zone. Here is a nice idea of how this clot looked like in the vial. And the vial is rather small. When we take the bacteria out, this is bacteria grown as seen, and they look like a flowery coal, like big clusters. And then when you stain for fibrin, it's really nicely coated. So we are convinced that we have a nice mimic. And here you can see the effect of rifampicin, though it is not that good. But we are working on how to set this up in order to facilitate that they also produce the toxins that I mentioned earlier. So that is, we are not there yet, but I hope that within half a year, we will be able to have a mimic. So we get a strain from someone who's bacteremic and has, of course, an endocarditis. Put the strain in our setup and then be able to support the clinician in making a decision on what antibiotics to be used. Hopefully not. So then, hopefully, we will be able to cure instead of to have an open heart surgery. Good. Now I need some water. I collaborate a lot with our cardiologists, and in my hospital, I should have moved it. So in my hospital, there's a lot of people that get an LVAD device, and these LVAD devices are so good that after 5 years, 60% of the people are still alive. So you don't need a heart transplantation, but they can. I'm talking too long.
In the presentation, can I ask question, Dr. Van Lommel? Yeah, because I see that you really focus to the bacteria itself, the cell, but what about the toxin? Because I know in a clinic we're using this kind of clindamycin, linezolid. This is one of the most important antibiotic when we deal with the toxemia in the clinic. Maybe you can also have to look to the toxin production instead of just focus to the bacterial number. What do you think about that, sir? Maybe because the picture like this, people will say, I won't use this antibiotic anymore, but that's wrong. We don't have anything else we can use in a clinic for toxemia. Linezolid one is the best, and clindamycin also the addition to other antibiotic. What do you think about this?
That's a very good question, difficult call. We know that certain antibiotics activate bacteriophages, for instance, which can be very nice because then the bacteria are killed by the phages themselves, by phage production. One of the problems is there's a lot of toxins on these bacteriophages. So, for instance, a PVL positive strain, you can activate with several antibiotics to produce more PVL. I think we need to know which antibiotics cause this and at what kind of concentrations, because there might be a zone where the killing is more strong and more quick than the toxin production. And I think you need to be at a level where you undertreat, so the bugs are still alive. They can get into some kind of defense, causing toxins to be produced. So potentially higher concentrations of antibiotics. I'm not a clinician, you know. But from bacteria point of view, I think that makes sense. So under treatment might cause toxin production up.
Thank you. If you might, I still have one opinion. The things that you saw, that if you compare what we found in the lab, that mostly lab results say that this bacteria is sensitive to this antibiotic, but when we go into the patient, the bacteria it just resistant to these things. Do you have a plan in the future to change this kind of conservative method that we're using in clinical microbiology now? Because I don't know if clinicians see this, they will say you're misleading us. I mean, the bacteria grow in human serum or human blood, but when we're testing in the lab, we're using like soya bean. That's not really natural in human. Maybe they become sensitive in vitro because they just doesn't like the soya bean. When you're adding antibiotic, they become die. But if you're using the blood, like you saw, they are more livable. What do you think this? Can you change the stigma among biologists? Because I mean, biologists is the one who have the great role for this kind of setting in vitro things.
I would say you hit the nail just in the perfect slam, and it's sitting in the woods. Bang. This is in my country. I have a hard time convincing our clinical microbiologists. The young start to listen or want to know the stories. The older ones don't listen. They just use their strategies currently available. And I think more and more the atmosphere changes, because also in the Netherlands, we have more and more problems with, and not even resistant strains, but difficult to treat infections, I always say. And yeah, then an operation and someone who has an osteomyelitis, debridement is, I think, the right word for this. And I think you know what debridement is, scraping. And can we do better? Like in the the endocarditis, losing your heart valves, please don't. Because then you're going to get an artificial heart valve, which is even more prone for bacteria contamination and accumulation and biofilm formation. So I often say that and the LVAD is a good example of this. It's very nice if you need a new heart and there is no donor heart available. Please put one in. But I can show you, we do this as I mentioned. Oh, this is a drive line on the left side normally. And in most cases, after some time, the right picture, yeah, this is an example of an LVAD drive line infection. So we give people a chance, though with the back door open, I always say. We need a way to keep the bugs out. Otherwise, they're going to be killed anyway. And when a person has a driveline infection, the list, the ranking on your possibility to get a new heart will go down. Yeah? So this is not just trouble. This might cause that you're not be allowed to get a new transplantation heart. Because what we saw, if you look at an MRI of this kind of, then the driveline looks like a neon tube. Huh? It's lit up like crazy. So what we, and if you don't mind, then I will continue. So what we do is we have a setup over here. We take these pieces of the driveline, and it's a special kind of Teflon. We just take pieces, and then we put in tissue culture media and allow it for 3 days. And every day the medium is changed. So after a while, then we look, and this is the first peak on the bottom. You can see that there's a nice biofilm being generated. And then we expose the biofilm to drugs. And that is the second part. And this is the control here. So there's no. And then after a while, we take the drugs out and we allow to regrow the bacteria. Because of course, that's also, it mimics the human situation. Huh? We don't constantly treat. So here is an example of rifampicin. This is the control without. And here you can already see that during the treatment, that the heat is different in comparison to the non-treated control. And at higher concentrations, then there's a lag time popping up. It's not for every experiment the same, but you can see that there is a slight delay at certain moments. And at a 0.5 microgram per ml, then there is no bacteria, and even you can really kill them because there is no regrowth when you take out the antibiotics. So we think that we have a nice way here to monitor these kind of infections on a driveline and potentially in the future help clinicians to come forward. And we have one case where we, my colleague Neliana Verkijk, who's also a former PhD student, treated the patients in different ways. And at a certain moment after a treatment, we could see that the bacteria increased. So when we mimic this in this way, we could indeed, using the calorimeter, show that under under Fluclox, it was rather low. But I think she used, I'm not sure which antibiotic she used. Then we could see that the heat was going up again. So you can really follow what is happening in vivo under this, using this setup. And now for the last thing, and that just only takes a couple of minutes, sorry. In one of the first slides, I showed you basically this part. Planktonic bacteria growing on the surface and then getting these kind of polyps. Well, nature. If you say something about nature, you're usually wrong because there will be when you look, this is for Azorius. When you look to Pseudomonas, Pseudomonas can grow like this, as I mentioned, but it can also grow in the liquid over here, like. There are even bacteria that can get loose from this, this kind of, and then they start forming hydrophobic molecules, and then they become floating on the surface. And here you can see examples of this. Here, this is it looks like milk that has been cooked wrongly. In this case, even here, I think this is an Acinetobacter. You can hold the tube upside down, and then it's strong enough to keep the liquid on top. Yeah? So these biofilms can come in whole different forms. And we are now looking also to Pseudomonas and Acinetobacter because these are also well-known nasty biofilm formation. But we need to come up with alternative ways to get the bacteria from a culture condition in our files. And using 3D printers, students of mine have been really playing nicely and came up with this form. And we can put these forms in a 96 well plate, grow the biofilm, and take them out. And then put them in the calorimeter. And then you can really harvest biofilms that are not just sitting on the source, but also moving around, also not moving around, but in plankton or even on the surface of the liquid. And so we have a whole different ways of, and of course, just use a 3D printer, a simple 3D printer, generate our own materials to work with Pseudomonas and Acinetobacter. A new master student will in September start working on this and look for a whole group of different Pseudomonas, if we can indeed do what we hope that we can do, and then of course determine the sensitivity for antibiotics in the same way as as I showed you earlier. So, and this is the last picture slides. We're able to monitor the behavior of biofilm-associated bacteria in a way we never were able to do. And in this way, we obtained a couple of eyes, a new set of eyes, allowing to observe in the unknown. I'm not working in a lab anymore. I don't like it, but I need to do other things. So people that helped me or people that I convinced, hey, we should do this experiment, are listed over here. And some of you, yeah, of course, Dr. Sultan is among them, and other PhD students that finished their thesis in my group. And of course, master students, technicians, and these are the funding and agents. Thank you for your attention.
Thank you so much, Professor, for that fascinating and enlightening presentation. We have certainly gained valuable insight today. So I came for the briefly summary. So Staphylococcus aureus is an important human pathogen that can cause a variety of disease, among which difficult to treat chronic infection. Prof. Willem and team tried to develop technique to determine the production of virulent factors of Staphylococcus aureus biofilm, which is associated with chronic infection due to their resistance against the immune system and antibiotic therapy, and discovered that Staphylococcus aureus already during the first stage of biofilm formation produce several immune modulators to mislead the immune system. And at this moment, Prof. Willem and team are looking for new ways to mimic the in vivo condition during endocarditis, osteomyelitis, and LVAD driveline infection, so they can, in the future, support their clinical colleagues in finding the right antibiotic therapy to treat patients, and to look for alternative therapeutic, including bacteriophage, baby, to fight this devastating infection. And of course, study pathogen and host interaction under clinically relevant condition. Okay. Now we have some time for question and discussion. If anyone has any question, please feel free to raise your hand or type them in the chat column in Zoom. We'll do our best to address as many questions as time allows, which is 1 hour for Q&A discussion. Please, maybe anyone who want to do research about biofilm? Yeah, Dr. Sultan? Oh, no? Anyone? Maybe in the Zoom meeting room?
The last one, please.
Yeah.
Thank you very much, Professor William. It's a very nice talk today about the biofilm. Yeah, I think we already have a discussion before via Zoom with Dr. Sultan also. I'm Aswad from Faculty of Pharmacy. I am pharmacist and especially medicinal chemistry. I interested in the biofilm and now try to make collaboration also with Dr. Sultan to do some small molecules that can inhibit the such as biofilm formation, and this to enhance the potency of the antibiotics. They're already resistant to the microbial, such as Staphylococcus and also the Acinetobacter. I see your experience with the biofilm formation. Actually, there are some key points such as the protein that can use by microbe to produce the biofilms, and actually that is my target to the to such as enzymes, for example, or other proteins that can help the bacteria to produce the biofilm. And what is the best target? That means what is the best target now that in your experience that can be used as a target of the molecules, like that small molecules, to help the to reduce the production or to inhibit the production of the biofilm. Did you find some protein target is now? Thank you.
Okay.
Thank you for this question. I'm thinking. If I knew, we would talk a different story.
Okay.
One of the problems here is redundancy. I think, and we know, as Aureus has a whole lot of ways to generate a biofilm. As I mentioned, the biofilm can be formed in fibrin. The biofilm can form in bone, and currently we don't know, are there different ways that these biofilms are formed, or that the book is using basically the same strategy, but using some small additions or changes in order to come up with something different. It could very well be that the basic regulatory systems are more or less the same. Yet the colors of the feathers, so the feathers are there, but the colors of the feathers are different. We hope in the near future. Currently, I ordered in the US, there is a transposon library of one Azorius strain. So every non-essential gene has been knocked out with a transposon. What I hope, for instance, in the endocarditis conditions and or the alpha driveline conditions, that we can select for regulatory systems that facilitate the formation of this biofilm. Are they the same under the different conditions? And if they are the same, then I can say to you, you should target this regulatory system. Huh? If they're not the same, then the trouble is is worse than I anticipate. We are looking here at the evolution of Azorius has been there for quite a while and knows how to cause issues in our system. So there must be different ways of getting that. And that's also what we bump into constantly. So in case of bacteria that are more accidental infections, and I think a lot of Pseudomonas infections are these kind of bacteria. And they're not sitting in us for a long time and then suddenly cause problems like Azorius. But they end up in us by accident and then cause a problem. And I think Acinetobacter is something like that as well. So hopefully for these bacteria, it will be a little bit more easy because they didn't have the time to develop different ways to cause the same problem. But this is pure speculation. Huh? I'm afraid in case of Azorius, this is a headache. Huh? Really, really trouble. And Staphylococcus epidermidis is there as well. Huh? One of these also, huh? this bacteria that are just sitting on us, huh? Dealing with us constantly, and then at a certain moment, cause a huge infection. While at most of the time, because half of us have Azorius in our nose. And are we suffering under this? No. Yet, in the end, I'm afraid I killed a lot of Staphylococci, huh? That I will be killed by them. Because I need some kind of medical treatment and I'm going to be neutropenic. And yeah. So I don't have a good answer at this moment for you. But this is, of course, what we need to find. And then we can put our targets on this system. And we can even come up with ways to screen for it. Because a lot of our, we have a GFP, you know, the use of GFP. So we can put a promoter in front of GFP and then, for instance, look for a strain or look for a treatment where the regulatory molecule is downregulated. Huh? And then we can easily, quickly screen. But at this moment, we don't know where to screen for. Yeah? So hopefully in 1 or 2 or maybe 3 years, we will be able to test this and look for the different regulatory molecules involved in the adjustment towards the human body. How do the bugs need to change themselves? And if we know this, I think this is fair game. Then we need to go after these. And furthermore, I like your approach because we have been studying also plants. Plants are always exposed to bacteria, sitting literally with their feet in the bugs. So, and they stand there the whole life. So they need to be able to deal with this bacteria, otherwise they are killed. And they do this already for quite some time. But a lot of people do experiments then with an extract and then show, oh, look, this extract is ethanol extract, this is active. And then they try to separate which molecule is it, and they don't find a lot of things. Because in the end, it looks like combinations facilitating this. And there's a very nice example, and it was a PNAS quite some time ago, and a PNAS. And then they could show that berberine is doing this. Yet, berberine binds to the, at a certain moment, there is a diaphragm being generated when a bacteria divides. And berberine prevents this formation of the diaphragm. So it's it's a very nice, it's a very nice molecule to target. Yet it needs to be there in high concentrations, and the bugs just pump them out, because bacteria have efflux pumps. But the plant came up with a good thing. They also generate an efflux pump inhibitor. So by just reducing the efflux pump activity, the concentration of berberine was already enough to kill the bugs. So what can we learn from plants by combining strategy, so different strategies to kill bacteria? So that is one other story that we are working on. And I'm not so much, I don't think that we're going to come up with the perfect antibiotic from plants, but maybe we can come up with the perfect strategy. Huh? What do we need to target simultaneously? So we don't need a lot of antibiotics, because I also know, and Dr. Sultan was talking about this yesterday, gentamicin, for instance, you don't want to use gentamicin in high concentrations. You're going to kill your kidneys, I was told. But if you can potentiate your gentamicin, then maybe we can reduce the amount of, but still get the positive effects of it. But how? Huh? And that's, of course, yeah, we need to search for ways. But then we need also conditions that are simultaneous, comparable to the human situation, instead of growing them in Müller-Hinton, which is milk, or tryptic soya broth. When does Azorius meet tryptic soya broth? It is not an intestinal, and for instance, in the Netherlands, we don't eat soya a lot. Huh? So, and still we have problems. Huh?
Okay.
Okay, Professor. They are very, yeah. Now my approaching now, yeah, find out try to guessing that such target, and then my calculation, yeah, using the simulation, molecular dynamic simulation that get possible before test in vitro, because of, yeah, if we test so many compound, it's gonna be costly. That's why, yeah, but the still trying to find out which one the potential target itself to can inhibit the production of the biofilm itself. Yeah. And yeah, they're approaching now. If they can, the simulation gonna predicting a good one, and then we test the by in vitro. So.
Very nice approach.
Thank you.
Thank you, Mr. Aswan.
Yes.
Okay, from pharmacy department. Okay, another question maybe? Okay, please, Dr. Dani.
Okay, thank you. So, your presentation highlight the role of immune modulators like SIN in early biofilm formation. Could you elaborate on potential therapies that might target these modulators to enhance immune response effectiveness against biofilm-associated bacteria?
There are a couple of regulatory molecules. So Azorius has global regulators that facilitate the production of, they look like some kind of switch, and then an RNA is being produced, and this guides the production of different immune modulators. And it could be that these regulatory molecules, so if we can target these regulatory molecules, they might just reduce the amount of production of immune modulators, making the bacteria vulnerable. I often say they just generate a fog around them. Our immune system cannot really penetrate in this fog. It cannot see anymore. Because one of the other immune modulators, for instance, bind to the C5a receptor, which is needed, of course, to find the chemoattractant. So the funny thing is, our immune system generates a huge amount of antibodies directed against these molecules. And still, it's not good enough. So why this is happening, and why the book can circumvent even the neutralization of, is it neutralizing? Maybe it is not. So we generate antibodies against these molecules, but still they can do their work in order to bind to neutrophils to prevent. So antibody therapy is not an idea because we already do this naturally. Because we have a system in my lab where we can determine the antibody response towards 70 antigens. For one of them, for instance, skin. So humans produce a lot of antibodies. It doesn't work. So we really probably need to target the regulatory molecules, facilitating this. And if you can target the regulatory molecule, you can of course also target instantly have the other reduction of the other molecules being produced by this regulatory. Yet, how to find ways to stop an RNA from being produced, or how to stop an RNA from facilitating, because these are regulatory RNAs, they have a certain form that opens up the mRNA from the target molecules, for instance. So you really need to go for maybe RNA targeting. COVID is not working like this, but yeah, maybe over there we can come up with. But immune therapy doesn't work. Until now, we collaborated also on a vaccine because I think 5, 6, or even more companies have been trying or are still trying to come up with an immune therapy. Until now, nothing works. Please realize that Azorius is producing protein A. And protein A is used by immunologists to isolate immunoglobulins. And this is on the surface of the bacteria. So they can just play with this. And then there are also a lot of molecules that just kill the proteases, kill the immunoglobulins. So I don't think that vaccination is the way to go. But maybe new molecules that can target the regulatory systems, or like, for instance, this two-component system that senses the antimicrobial peptides outside. Can we block it? But we shouldn't activate it, because then you even get more problems. So can we find a way to target the response of the natural response toward antimicrobial peptides, making them less positive, so our antibiotics can do our work? I think these are possibilities. Fantasies, maybe, but maybe also with the modern ways and making all kind of models coming up with. But you also need to get these molecules there. And how do you do this? Because biofilms also tend to be at places where blood is not so much, for instance, in bone. Yeah. How do you get your target molecules over there? So maybe we need to open the wound and put the components on top of it, something like that. But yeah, it's going to be a difficult call. Or maybe we should just leave the biofilm and try to make it sleep. Because, and I'm not sure if you know this, but I know stories that Azorius can be just sitting in bone for 20 years, and then suddenly the person gets becomes bacteremic, and then they find that, for instance, they broke their leg 20 years ago, and then you end up having a bone that's totally crowded with Azorius. Why didn't it become a problem earlier? The bot was sitting there. So can we get them back to this hibernation form? Is that a way to go? Of course, you want to get it out, but if you can't get it out and someone is losing his foot, maybe by just getting it in again and silent is a way to go. I'm a biologist, yeah, so I think, and that's why I need to talk to you guys, because you're going to say, hey, this is not possible. You cannot just add this antibiotic because it's going to kill the kidneys. So please help me.
Okay, thank you, Dr.
Okay, thank you, sir.
Okay, maybe from the intern resident or clinical pathology resident have a question? Doctor? Or from clinical nutrition resident?
Yeah, maybe from clinician, because we have also the real clinician here. Microbiologist is not really clinician. It's just like a addition for clinician. But maybe from infectious disease, maybe they have their own opinion, especially the use of drug in the biofilm.
Good morning, Prof. I'm Suryana. I'm trainee of tropical infection in internal department. And I want to ask you some question. In our experience in hospital, in inpatient hospital, we always face many cases of methicillin-resistant Staphylococcus aureus. And in MRSA patient, especially in patient with device like endotracheal tube, central venous catheter, or double lumen catheter for hemodialysis. And many factors that can influence the difficult to treat this infection, like albumin levels or comorbidity, and etc. And my question is, can we give, in your opinion, can we give the combination of methicillin-resistant Staphylococcus aureus patient vancomycin with your presentation in the first case we met the patient of MRSA? Because especially in Wahidin Husodo Hospital, as a tertiary hospital, many factors that happen, like we can replace the device because the condition of the patient not stable, like the ETT or central venous catheter, or we can exclude the infection, the focus infection, like infection in eye, in patient in malignancy, we can treat the focus infection. So the risk of MRSA is very high. Please give your opinion. Thank you.
Yeah. I'm not sure how to respond to this. This is indeed the trouble. And how to approach this. Maybe we should make a mimic of the different. There are, yeah. I'm afraid this is more for a question for a clinician. Huh? I'm afraid that, for instance, an MRSA on a biofilm, that is your worst scenario. MSSAs can, for instance, a little bit have some possibilities, but the MRSAs, you even have more difficulties. What I recently, I stumbled into literature on MRSAs being sensitive to fluclox, despite being an MRSA. And this has to do with, in humans, baking soda, it sounds a little bit crazy, but in human, baking soda is the buffer that is in our blood. And it has been discovered that a certain set of MRSAs in the presence of baking soda are less sensitive to, less resistant to beta-lactams. So we are currently trying to come up with ways to figure out which MRSA has this possibility. Not all MRSAs. It's particular from a certain genetic background, CC8. These are sensitive. So in combination of baking soda, they are sensitive. And what they did is they took bacteria media and added as a buffer these baking soda in human concentrations. And then they could show that they become sensitive for beta-lactams. So potentially, we need to come up with ways to test this in the future and to see which patient has a strain that is an MSA that can be treated with beta-lactam. So you can at least approach these. the real MRSAs on a biofilm, at this moment, I don't have an answer. I really don't have an answer. With the present antibiotics, you know better than me, then there often is no answer. So we need to come up with alternative ways to fight them, and hopefully we can find ways to, like the example that I mentioned in case of berberin. Berberin is the active component, but you need to add, for instance, an efflux pump inhibitor to get high concentrations of berberin inside the bacterial cell. Can we fight MRSAs in the same way in the future? Using other ways in combinations, and maybe come up with inspiration from plant world to fight this. But like I mentioned, in case of baking soda, it sounds like an illusion, but it's really because in the Müller-Hinton, there is no buffer. While in our bodies, there is a buffer, of course. Like, because we, and that is based on CO2, of course, which causes to make baking soda in our bodies. So testing in vitro and testing in vivo, there are examples where they are more sensitive in humans, actually, than you would if you look in vitro on a plate or in your Vitek. So this might be a possibility, but that's, I don't dare to say more. But we need to come up with strategies to quickly filter out which strains can be treated with beta-lactams, although there are MRSAs.
Okay.
Maybe as addition, sorry to interrupt, as addition from Clinician Association of, I forgot, American Society for Infectious Disease. In the case of biofilm, there is no such thing we call MSA or MRSA because all of them behave the same, like Dr. Van Wammel saw here. So the protocol now, they say maybe they say that you can use addition, a combination with rifampicin. For example, if you have biofilm like LFAT or something with the PPRA can authorize you directly to use vancomycin, but you have to adding rifampicin. But the question now, we are in Indonesia, we have a problem with tuberculosis. We cannot just give people rifampicin, and this is no solution until now. But if you want to add rifampicin, it really depends on your responsibility because there is no agreement in Indonesia yet. Even I think in Europe, yeah, Dr. Van Wammel. Yeah, I remember this have discussion with Milian, but what we have now, the best way to tackle anti-biofilm is rifampicin. But you see, in the setting after, if you give rifampicin alone after 24 hours, after 10 hours, I guess they up again, and that's not good at all. And one thing you make sure if you want to start this combination, you have to make sure that patient doesn't have aureus in their blood. So there is no aureus in their blood. So if you take one case with biofilm, you have to make sure there is no aureus in the blood. Because if you adding directly the rifampicin, this is going to be really trouble. It will become really resistant. The plantonica one really sensitive, reacting really bad to these things. So you have to give, like, for example, I think Meliane in Netherlands, they give flucloxacillin for 2 weeks. And then they check the bacteria from blood. If negative, then they can start the combination of rifampicin to treat the antibiotic patient. But it really depends if it's already entered the bone. If you even if you use like a 1 kilogram, it's not gonna work. You have to do the cutting out. That's too bad, but that's the reality. I think if it's already entered the osteoblast or osteoclast, there is no cure for at this moment. Thank you.
Thank you, Dr. Sultan. How about the answer, Dr. Suryana? Are you satisfied? Okay. I continue to maybe another question from resident or maybe participant in Zoom meeting. Silahkan dok, kalau ada yang mau bertanya di Zoom, di ruangan ini.
Ada mungkin pertanyaan dari kawan-kawan IPK mengenai teknik dan sebagainya. Ayo, jangan hesitate, ayo bertanya saja. Silakan.
Ya, yang dari patologi klinik, monggo dok.
Dari mikrobiologi residen mikro, ada pertanyaan? Any question from resident?
Mungkin yang mau buat penelitian.
Yeah, especially to one have doing their thesis experiment now. Dari farmasi, mungkin S2. Ayo, tanya saja. Well, Indonesian always shy to ask questions. Even for Sulawesi. Please don't hesitate. You can ask question directly to Dr. Fanamal. He's a good guy. 10 years we are together. So 10 years, right? We're working together. It's a good memory.
Indeed. That's why I'm here.
Ayo kawan-kawan, ada yang bertanya? Mungkin ada pertanyaan? Ya, kalau tidak ada mungkin tutup saja.
Okay, if there is no other questions, so this is time for us to closing this event. As we wrap up, I'd like to express our sincere gratitude to Prof. Dr. Willem for your time and expertise, and a big thank you to all of you for attending and participating in today's guest lecture. We hope you found the session informative and inspiring, and have a wonderful rest of the day, and we look forward to seeing you at our future events. Thank you and goodbye. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih. Thank you, Prof.
Terima kasih atas partisipasi semua, terutama yang di Zoom. Mungkin kawan-kawan yang dari foto bersama Prof. Willem, silakan.
Dok.
Tanda tangan boleh juga, Dok. Untuk akreditasi penting. Ayo, silakan, Dok, silakan.
Iya, boleh mau foto bersama, silakan, Dok.
Please.
Ayo, Dok, monggo sama Prof. Kalau
ya,
mau
silakan
foto.
dari bagian patologi mungkin dulu, silakan, Dok. So they want to make a picture with you, Dr. Willem. They are from clinical pathology, Prof. I think you call them biochemistry department, sir.
You want me to remain seated?
Fotokan, Herut, fotokan.
Okay. Kayak tidak seimbang gitu. Geser-geser boleh. Satu lagi supaya seimbang. Oke. Satu, dua.
Kawan-kawan dari SP2 penyakit infeksi, silakan. Once more, sir, from clinical infection to more, I guess. Pak Topik, sertifikat peserta udah ada. Kayak bagi peserta lain yang mau ngambil sertifikatnya, Dok, ya bukan SKD ID ya, tapi bagus lah buat BKD, lumayan. Silakan, Dok. Dua minggu. Kamu dibawa ke mana? Iya, dia mau ke Bira nanti hari Jumat ini, bakso. Ya, kawan-kawan dari farmasi mungkin dan S2 silakan, Dok. Ayo, Pak Aswat, farmasi silakan. Bantu Pak Taufik ya. Yeah, we love pictures and we love bule. Hah? Kenapa?
So how do you want? Like standing? No, but standing or sitting? Yeah, no, but standing or sitting?
Taufik, linknya perlu izin kayaknya. Nggak bisa langsung. Link sertifikatnya.
Mana aja hasil AGD kan ya?
Oh, dari mana dong? Dari ini.
Nggak bisa. Tunggu ya, diperbaiki.
She knows how difficult it is to work.
Yeah, we try now using the this is cinnamic acid derivatives. Yeah.
So I see Holland Bakker.
Ayo dok, silakan menikmati kadarnya. Mungkin kawan-kawan di Zoom sudah bagus linknya, tutup Pak Taufik. Sudah bisa. Sudah bisa. Cukup sekian.
Yeah, I will.
Yes.
Tulips. I just gave tulips.
Foto sama mami, ayo.
Aha.
Foto kan mami itu, foto mami. Bisa?
You want me to sit or do you want to stand?
Yeah.
Do you want me to stand or want to sit?
Okay. Okay.
So you feel
Sahabat dok, yang linknya masih diperbaiki, soalnya perlu authorize.
That's Erasmus Hospital. It used to be Dexy. Yeah, I was there in Dexy Hospital and stay in the nurse house. Hi.
so that was in the 60s.
Yeah, 86.
86.
It's pretty old time.
And I was just born.
Yeah, just born. Clearly.
Yeah. Oh no, 86. Of course, no. 6. Sorry,
That's when
I'm wrong. I was
Dr. Dehner,
Yeah.
Dr. Dehner,
Dehner, yeah.
my supervisor.
no, 86. Then I started studying biology in 85. Sorry, yeah.
91. In 91, I was in Amsterdam for PCR. Maybe I am the first one studying PCR in Indonesia at the time.
Interesting.
Because
And what kind of PCR did you use?
Yeah, the Royal Tropical Institute for lab sourcing.
So perfect. Yeah. So I started when 91. I started my PhD in Utrecht in clinical microbiology department. So you have been often to the Netherlands.
2 times. I'm the oldest clinical microbiologist in Makassar. And maybe one.
Ya, silakan di-scan supaya bisa dapat sertifikatnya dok buat BKD.
But you have a lot of experience.
Udah bisa.
You have a
Belum
lot
bisa?
of experience.
Belum bisa Taufik.
That's important. And I'm not a clinical
Ini
microbiologist.
sudah bisa yang lain. Kawan-kawan dari patologi ada?
So treatment is not my.
Oh, masih belum.
But of course, we try to understand how the books are behaving. So can we then predict and help you make a decision?
The other bacteria have the same pathogenesis. Yeah. I don't have to.
Yeah, I'm going to have a look. What is inside? The Holland Bakery.
But
No
this is
kidding.
Indonesia.
Yeah, that's what I. Thank you.
Dokter Dani bisa dapat? Bisa, Dokter Dani? Scan anunya, sertifikatnya. Yang lain ada yang bisa. Coba tutup dulu browsernya yang terbuka tadi, baru scan ulang. Bisa memang.
I was the sorry. Hidden, yeah. But the people all in me, older than me, can speak Dutch very well.
Yeah, that's what I understand.
I will
Bisa?
just speak a little.
Dok, boleh ditulis emailnya dok, kami kirimkan masing-masing nanti ke emailnya.
Dus u kan Nederlands spreken.
Yeah,
U spreekt Nederlands.
my brother,
Ah.
my old brother.
Ja, ja. Ja. And you were born in Makassar.
Makassar, yeah.
Yeah.
Ada absen dulu. Tapi dok, boleh kita kirim tulis saja email tak nanti di depan di listnya. Sudah bisa? Nanti kami kirimkan ke email tak masing-masing.
Yeah, we hear a lot about Indonesia, and I've never been here, so
Yeah.
it's interesting to come to Indonesia. And of course, I collaborate.
This is your first time
Yep.
to Indonesia?
Yep.
Now, how long have you been? How long you are?
Linknya sudah bisa dok dipakai. Silakan. Bisa di scan sekarang. Itu aja tadi.
And I worked, of course, I'm already more than 10 years.
Yeah.
Did it first this month.
Aman kan?
My
Ya.
department.
Silakan dok. Di-share saja dok. Kami akan aktifkan selama 1 jam ke depan, setelah itu tutup.
We worked a lot together. He has a very nice contribution to my group.
Yeah. Thanks very much.
Yep.
Because he wanted to do the collaboration.
Yeah.
Is it possible for him to get like an invitation letter to your lab for live?
Of course.
Yeah, actually, I have an opportunity this year funding from the university. Yeah, maybe I can for the what 2 weeks.
2 weeks. What do you get with 2 weeks? Can I get it in 2 weeks? A month.
A month.
It's quite a complicated procedure.
Oh, really?
Yeah.
In 1 month.
Yeah, because you need to learn a lot for
Of course.
it's only 2 weeks, I don't think it's easy.
2 weeks is not enough.
Not enough. Maybe you can just learn crystal fire and done. No, if you want.
Okay. Well,
What do you think? So
what do you think?
No, I don't work in the lab anymore.
Oh, really? He's the boss.
I talk.
But you're the boss.
Yeah, I know. But, it depends on what you want.
What do you want to do?
Actually, first our students were because now we try several compounds in biotech. Maybe it's better if we found that test in your with your method.
1 month.
And then it just depends on what you need to do.
Yeah.
So then we can make a calculation of what is reasonable. It really depends.
You can also leave your things after 2 weeks. You leave your compound, I think they will continue, I guess, if they have time. If they have time. Is it possible? Because you can ask me.
Actually, yeah, I never been in Europe. Yeah, I graduated from Japan. Actually, I don't know how to.
I know you can work like for 24 hours. I think it's enough for him. Maybe for the beginning, so just give him 2 weeks.
Actually, the funding is not.
It's not that much.
It's not that much. Maybe yeah. Just I can get 2 ST. Yeah, that's why if maybe I can for 1 month. But the most problem is in
Maybe the beginning is just 2 weeks because it's quite expensive.
in the beginning.
Yeah, of course.
Is it possible to do something?
You're welcome. That's one thing. Question is just how much, what do you want to do? And reserve the machine. And in order to do it, you need the hands of, for instance, Cornet.
Yep.
That's my technician that is dedicated, devoted to do these experiments. And he can show you how to do it. And if you want to do it yourself, at a certain moment you can do it yourself as well. And then it's just a matter of how much, what kind of book you want to study. Did we already have a setup to work with? Then it's quickly done because then we just take care of it.
Tapi dok, ini acaranya sudah selesai, resmi ditutup. Jadi kalau ada kegiatan lain dari teman-teman residen lain, monggo dok, silahkan boleh meninggalkan. Absen dulu dok, sudah bisa.
Because you want to establish your policy, and then you need a certain amount of energy. So you need to determine when is this a reasonable amount.
Because you know that kinetic works. Kind of thing.
If you're too early, then yeah. So that is important to figure out how to do that. In case of Azorius, we had everything. And you can even play around with it.
Computational docking and dynamics, molecular dynamics.
He always playing with molecular docking, so it's quite good.
Oh, and
yeah, I try to find out that several targets for the if it's possible, especially for the unsigned, because we work with the protein or the small molecule. That's why I try to find the way for the biofuel information.
At times, I think I already tried to do one of your compound. I'm afraid your compounds not really kill the bacteria, but playing around with the toxin production. So maybe you can use the GFP. So that's pretty.
Oh, my favorite compound. Yeah, actually, I synthesized the problem by myself. I just combine the two compounds. Yeah, because I can modify the whole molecule. Yeah, I'm medicinal chemist.
Have affinity to the oils.
I like that.
Well, do it the collaboration.
Yeah.
But you
talk about berberine. Berberine actually they have positive charge. Maybe. Yeah, better to find the compound that has the positive charge.
Funny, there are conditions when positive charge is benefit.
Yeah.
There are conditions where it has been described that if bacteria will acidify their surroundings, if you buffer it, then this acidification is important in order to generate ATP, because they have the proton motive force. And if you frustrate the proton motive force, then the bug will try to come up with an alternative, and it will start to spit out potassium. But the potassium is protecting the bug from getting positive molecules in, because the potassium is buffering the negative charge of the DNA and RNA. So you get a gradient over the membrane for charge. So if the bug starts to spit out in neutral conditions potassium, then positive antibiotics get in. So there are conditions. That's why I think we need to really figure out how deep condition is. And baking soda is doing that.
Of course.
And baking soda is, of course,
baking soda, of course.
But it's also part of our buffer in our system.
Of course.
It's the same. In our blood, baking soda is the buffer. And that's, of course, due to the RCO2 concentration. Because there's a whole dynamic going on. And so, but he doesn't want to use what is it? Gentamicin in high concentration, because you're going to kill your but for certain conditions, I wouldn't be surprised if gentamicin is helpful.
Yes.
So can you, in a bone, for instance, locally, get the pH down? Sorry, the pH up.
Yes.
And then, so I wouldn't be surprised if there are possibilities. Going back to what is the future conditions? So how does your molecule look like? What do you anticipate? And what is a physiological condition where this might work? Because if you are in endocarditis, then you're constantly buffering, because in our blood there is so I wouldn't be surprised over there that especially on the surface of the body, the pH is really neutral. Maybe inside it might be a little bit acidic.
Yeah.
Because, but if you can work around and start killing the outside from outside in, you might. So it is. I'm afraid it all depends on what you want to study.
Yes.
If it's going to be related, reasonable in the context where you want to use it. But we can, of course, start easily. Just use.
Just know how it works. Because, actually, this is interesting. Actually, I need that's for you. You can and submit the letter to
talk with my chair of the department, but I'm sure that he will agree. I will write together with him, because I cannot write it. I need to ask my chair. But we will, and then, because I will make space in my doesn't matter. It doesn't matter. But then, of course, we also have, what do you want to test? Because we have, I didn't mention that here, we have GFP, like Roshan mentioned, we have GFP promoters, and for different regulatory systems, we just put the promoters of this regulatory system in front of you, and then you can grow the book. And when it becomes free, you know that the regulatory system is on. And then you know, for instance, alpha-aminoacid is being produced. Or we can also have promoter of alpha-aminoacid. Put it in. And so at different levels, you can have a look if the molecule is playing a role on regulatory systems or on the final protein that's toxic. And if you know, of course, where you target, and we can look to have a readout system that is different from the Casimir. And the GFP, I'm going to share. But then you need to, these are genetically engineered as always. And they are a difficult thing. I'm not allowed to just send it to you. So then you need a special lab to work with it. That is far more easy, because then you only have to look, is the book getting green? And is your treatment reducing, for instance? That is more easy.
Because consider time, maybe it's a bit easier.
And then you can really go for a whole lot of different conditions, because you can do this in 96 well. And just detect what condition has an effect on your. But of course, then choosing the right strain is. Because if you choose wrong and you don't see anything, then you say, "Hey, there's no difference."
But the other maybe.
So, but of course, we can also check for different. Yeah, there's a lot of possibilities. Sorry, my mind. Let me check. What is this?
Yeah, that's right. Sure. Holland. Holland bakery. This is first time. Indonesia?
Indonesia.
Wow.
Here I am.
When you arrived? Last week,
yesterday morning. Yeah, at 2 o'clock in the morning.
Maybe still jet lag like us here in the Netherlands around 7.
So very
6 hours. Yes, 6 hours. And when is in University Hotel?
No.
Oh, no.
I don't think so.
Oh, no. Outside, near the sea?
Yeah. But it's in town.
I think you are in because you have the University Hotel now. Because Loket is is it like countryside? Maybe that's
I don't mind being in the country. Not so much. I'm a biologist.
I'm chemical biologist. Yeah, I'm working chemical biology. Yeah, especially for protein. Yeah, because I work to let's say how to be. Actually, in Japan, I'm working how to determine the binding site of the protein. There can be targets for the drugs.
So for the binding site?
Binding site. You know, the protein or amino acid surrounding the binding site.
So you can really compete with.
Yeah, for everything.
So what did you do for where is your targets currently aiming for?
Actually, in Indonesia, I cannot do. Yeah, because of it's quite difficult. The method is quite difficult to do in Indonesia. That's why now I will be changed because of Actually, I'm chemical biologist. That's why I can modify. I can obtain compound from the plant and then modify them, and directly make in vitro say that. Yeah, just like that, because of such kind of molecular thing that I did in Japan previously. It's quite difficult that's why now I try this. Collaborate with find out I can find out the compound from plant or synthesize or modify and then directly. Now, my target is not to find out the antimicrobial because it cannot be very difficult. That's why try to find out the other things that related to like resensitizing the microbial resistant. So now target first by flux pump. But before that, make like calculation.
Can you prove that this is an efflux pump inhibitor?
I can take simulate first.
Yep.
Like for example, target is NOR-A, for example, NOR-A. Then I make
I think I have a NOR-A mutant.
I just found the PDB from the I just can found PDB from the PDB. And then I calculate like simulation first. Simulation. If the result that's why I screen I did.
I mean, it's important to you to know that I think I have a NOR-A mutant. You understand what I mean? I have in Azorius, NOR-A is playing a role, of course. I think I do have a knockout for a strain that doesn't produce NOR-A. So using this kind of strain, you can show that it's NOR-A specific. So you can, because there's a lot of efflux pumps in Azorius, and different efflux pumps, there are assays to do this. You can add, for instance, I think ethidium bromide.
Ethidium bromide.
And this kind of things. And then you can really see nice zones of inhibition or activation of these kind of things.
I will. It just maybe this month. Because I already have the result of the simulation. The consultant
This doesn't look like it's
this is sticky rice inside.
Okay.
Sticky rice. This is sticky rice, and then wrap it with the banana leaves, and then bake it, grill.
And do you eat the
No. This is just cover. Okay. Just cover. You can open it. Yes.
There's
you can. Dangerous. How about Indonesian food?
Nice. Until now. As long as I don't get sick. Do you think it's wise to eat?
Yeah, why not? That's only like a
fried.
like a sushi.
Okay.
But the better version, it's just chicken with sticky rice. It's really nice one, so better than
This is shredded chicken inside and
It's
also the sticky rice.
It's using Dutch words.
Iya, bisa. Nanti tinggal dikasih saja. Dikasih pokok contohnya. Nanti tinggal dikasih. Saya butuh kasih.
Oh, ke lab dulu, nah, gua harus dicek dulu Aureus. Before you go to Netherlands, we have to check you for
Yeah,
Aureus first.
Of course.
We have to check your nose and your mouth.
Because of, actually the student can do that for the Aureus. And do that. And I already have a compound from.
You can bring to Netherlands, I guess.
The compound from Japan.
Oh yeah,
they produce that. Yeah, we have also collaborated.
Yeah, we're working with the small things like MIC.
And there are compound produced by actinomycetes. And then they produce.
Herut mana tas merah yang tadi di situ? Herut sini dulu.
The compound can isolate. He will check. He will check it also.
To bring to Netherlands. It's gonna be like a one. Wow, that's nice. Resin.
Jadi apa kira-kira apa dia bekerja lagi?
Biofilm, antibiotik, apapun yang bekerja. Oh, di sini saya mau bawa ke jadi bagaimana? Tergantung saya mau bawa kemana juga. Kayaknya ramang-ramang saya keliling sudah jadwal. Pulang. Kau Mi, kapan kau mau pulang?
Oh, okay.
Mungkin
we can go to the island. Actually,
you want to visit islands?
You like
offer
snorkeling?
you. Yeah, he loves that.
He loves snorkeling.
But this, just tell me the schedule because I need to accommodate also. He even doesn't know what his schedule is. Yeah. Which pulau mana kau mau bawa?
Sama Lona and Kodingarek.
Oke.
Kamu Penambungan.
Kodingarek. Penambungan kau bawa jauhnya.
Kodingarek itu keren for snorkel. Not for diving, just snorkel. Just snorkel.
That's actually nice. But you won't find this in the Netherlands, so the Holland back. If you come to the Netherlands and looking for sticky rice, there was what do you mean?
But they have it in Albert Heijn, sir. They call it onigiri. That's like a
onigiri.
Onigiri, but the taste is terrible.
They
have it's not lamparsa, but they have the Japanese version
in Holland.
Yeah, really? Albert Heijn.
Onigiri like that.
But it tastes really disgusting. I tried that. I don't know. It tastes like a plastic. I have to say. I don't think that's.
He knows better than
I don't think that's Japanese food because it tastes like a plastic.
Because this tastes really nice. So
I know him since I was undergraduate. We are in the same university but different faculty. He's in the medicine and I'm in pharmacy.
Yes.
And now we are working in the university together again.
even I am the chair of the department. I don't have this one. I never have this one. So it's not fair. They never get me. Seriously.
So what is it?
Why you don't know that? You're the head of.
Oh,
It's pretty cool. Kita plakatnya, Mami. We never get this one. Yeah, it's a special for him. Oh yeah. I don't have one, sir.
Oh,
you have
nice.
the diamonds. I mean,
Wow.
there's always different levels. This is the blue one, and that's
Oh yeah.
yeah, that's an important one. My goodness,
A lot of things.
I'm gonna. What is this?
We're still waiting the team for the lunch. We already prepared downstairs, but you can sit down now for a moment here. Should try this one, sir. You know this?
No.
It tastes like lychee. I don't know, lychee. Do you know lychee?
Yeah.
It's much better, I guess. It's much better.
Thank you for helping me out in the last question.
No, it's all right, sir.
No, because that is not a question that I should answer.
Nice. Yeah, I shouldn't do that, but yeah, it's okay.
No, I mean it's a clinical microbiology department question.
Yeah.